Okay, we are now online. So I think that we can slowly start. So good afternoon, everyone. Today we are pleased to have Judy Kim, a speaker of the seminar series of the Brazilian Physical Society in the area of atomic and molecular physics. Judy Kim is currently professor of chemistry and biochemistry at the University of California, San Diego. She studied at the MIT for her bachelor and master, and then she moved to Berkeley, where she received her PhD in 2001. She also had postdoctoral appointments at Berkeley and Caltech. Judy Kim's research interests are at the interface of biophysics and chemistry, and the current research directions of her lab include spectroscopic studies of membrane protein folding and dynamics. The title of today's colloquium is Singlets, Doublets, and Triplets in a Protein, a study of a biological electron transfer reaction. YouTube attendees can interact with the speaker by asking questions via chat that will be read by the committee members. So please, Judy, you can start.
Great, wonderful. Well, thank you very much for that introduction. And as mentioned, thank you all. My name is Judy Kim. I'm from UC San Diego in the Department of Chemistry and Biochemistry. It's a real honor for me to be here today. Very important. I want to thank the organizers, Tommaso, who invited me, Sandra, my good colleague Silvio, who I understand is traveling, and for all of you for joining me. Good afternoon, good evening, good morning, wherever you are. I know that we all have busy schedules, so I'm really excited to be able to share with you our research. And I'm coming from San Diego, a very lovely city. You can see our campus here is less than a mile from the beach, so very easy to go and get some sunshine while we are doing our research. And if any of you find yourselves in San Diego, please visit our wonderful campus. We have some, in addition to, of course, research and science, we have some very nice artwork. This is one of my favorite art pieces, which is a house that is hanging off the engineering complex. This is when they were installing it. It's a real little house. We'll go inside and I will give you a tour of our campus and our nice pieces of art. And as we, in case I don't get to this also very important slide, which is to thank the students and the agencies that have funded the work. All of the work that I'll talk about today is really Joel Rivera, Visha Jana, Christina Trin, Justine, and Ignacio. They've all participated in some form of the talk that I'm going to give today. And then some previous members as well as collaborators, Greg, Hannah, Chris Lee. They were all very important for the work. And then there's other members of the group who have contributed or participating in other projects that I certainly want to thank for their efforts. Funding from National Science Foundation, National Institutes of Health, Department of Education, all in the form of research, as well as really generous student support, training grants, as well as fellowships. So again, gratitude to all the folks who have made the work possible. Okay, so today we are going to go on a little journey about electron transfer and biophysics, as well as thinking about how it is that protons can do one of the easiest reactions in life, but yet it is actually quite a difficult reaction in many ways. So I'll start out here with on the left side, which is what's known as the tunneling timetable. And this is a plot of the time it takes for electrons to travel certain distance, depending on the medium. So whether it's traveling through vacuum, through water, or through proteins, which is the focus for today. All the dots here, so this is a graph that comes from Harry Gray's group, and much of these dots, or all of them, are from his group. What they have done is labeled proteins, various proteins with various labels that have distances that are known. They determine the amount of time it takes to travel from certain points, and that is what this scatter plot is here. The key thing, if you notice, the y-axis is logarithmic, so it is not a linear graph here. And so this highlights the importance of short-range ET events and long-range reactions. So for example, if an electron needs to travel 20 angstroms to get from point A to point B, it might take, say, 10 microseconds. And if you just cut that distance by half, you gain 6 orders of magnitude in terms of time. So the efficiency, of course, is dramatically increased simply by reducing the distance. Of course, this makes sense, and nature does this, otherwise we would not be alive today, is we have many molecules, cofactors in our in biology that enables electrons to move long distances for respiration, photosynthesis, etc. What are some of the typical centers for, that act as these intermediate hopping spots so you can break down a 20 angstrom step into 2 angstrom steps? Lots of transition metals that as you might expect, such as iron, copper, etc., as well as organic cofactors. So there are organic molecules that can be relatively easily reduced and oxidized that participate to enable long-range electron transfer. Our group is interested in amino acids that can act as intermediates. So, and what I've drawn here are 4 of the 20 natural amino acids: tryptophan, tyrosine, cysteine, glycine. These have all been studied in terms of their role for redox chemistry and electron transfer in proteins. So it's nice that you can actually use an amino acid for electron transfer. It's not something you think about typically, because when you think of electron transfer in proteins, the metal centers come to mind, and for very good reason. They're much easier to reduce and oxidize. They, as long as you can install them in a protein, they're very valuable. The benefit of an amino acid, of course, is that they're already built into a protein. So a protein does not need to build in this structure to support a cofactor like a metal center. They already exist. The drawback, of course, is these are organic molecules. Their redox properties may not be quite as easy to, their reduction oxidation may not quite be as easy as a transition metal. And you see many of them have protons. So if you were to make a radical on the tyrosine or cysteine, glycine, or tryptophan, you can have charged or neutral radicals. But they're really, being discovered as more prevalent in proteins. And so our focus here is on the tryptophan radical. Why the tryptophan? Really, it's because it's one that hasn't been studied very much, certainly comparison to its partner, for example, the tyrosine radical, which has been studied earlier. And the tryptophan radical, depending on whether you think they're old or not, the first radical was reported only in the late 80s. This was from EPR measurements from Brian Hoffman's group over at Northwestern, and they reported a cation radical in cytochrome C peroxidase. So this is when the first experimental study of the radical, of a tryptophan radical, was reported. But even before that, there were earlier thoughts and proposals of the tryptophan radical, and this was by a great theoretical physical chemist, the late Klaus Schulten, in the late 70s, with his co-workers, proposed that the tryptophan radical was involved in long-range migration of birds and insects. This sounds like a complete disconnect, but it's not. And I love this story, and so I just want to take 30 seconds to describe this. I'm here over in California, and we have, if I go up to the Northern California or Mexico, there's these beautiful butterflies, monarch butterflies, that travel between Canada and Mexico, and they go back and forth. This is their cycle. This travel takes longer than the lifespan of a monarch butterfly. So even though a monarch butterfly has never been to a particular location, it knows where to go. How does it know where to go? So it was proposed by Klaus Schulten and coworkers that these butterflies have what are called magnetoreceptors. In other words, they can see the magnetic field because of organic radical pairs. It took decades later, so about 36 years later, another group detected magnetoreceptors in the antennae of these butterflies. And so now the question is, what is the chemical and biophysical basis for these magnetoreceptors? And sure enough, it's been proposed that they involve tryptophan radicals as well as radical pair interaction. So in this case, in terms of the butterfly antennae, there's a protein called cryptochrome. Cryptochrome has a flavin cofactor, and it has this triad of tryptophan. Photoexcytoflavin, an electron is ejected, filled in, electron transfer goes from tryptophan to the flavin. A second tryptophan fills that hole. A third tryptophan fills the hole of the second tryptophan, and you're left with a cation radical on one end and the flavin anion radical on the other end. This radical pair can be a singlet or a triplet. So both spins can be paired, which is the triplet form, or they could be anti-parallel, which is the singlet form. And that conversion is very sensitive to the external magnetic field of the Earth, and therefore the signaling state of the protein is sensitive to the magnetic field of the Earth. So this is the proposal of how this works. Now, since then, and since the tryptophan radicals have been reported in the literature, there are, of course, many other examples of where the tryptophan radical is involved. And I'm just putting a few examples of enzymes here where all have tryptophan radicals: ribonucleotide reductase, which is involved in RNA to DNA chemistry, cytochrome C oxidase, cytochrome C interface, so electron transfer, difficult chemistry, which involves the tryptophan radical, and of course cryptochrome, which is what I just showed you. And then a very related, structurally at least, a related enzyme called DNA photolyase also has this triad. I like this pair here because when I was reading up on how it is that these two enzymes have such similar sort of similar structures but different functions, and it turns out that DNA photolyase is a very ancient enzyme, and it was important back when there was no ozone layer protecting life. So DNA was constantly being damaged. This enzyme would repair it. When the ozone layer appeared, there was not as much need for DNA photolyase, but instead now creatures needed to kind of understand, needed to develop a method for circadian rhythm. In other words, they evolved the photolyase into cryptochrome because with the development of the ozone layer and then early life, the goal is to sort of avoid daylight. And so you needed a mechanism for circadian rhythm. I'm not describing this quite as well as I'd like to, but it's just such an interesting connection between these proteins and enzymes. Back to the goal here, the purpose here is to illustrate that, yes, these tryptophan radicals are quite prevalent. We don't know a lot about them, and so our group has set out to study these radicals in the context of electron transfer. So let's look a little bit at the sort of the chemistry and the physical chemistry of how it is that the radicals are formed. Here's the side chain of tryptophan. This is called indole. So I'm just going to denote it TRIP-H. So it's got two rings. How is it that it's going to give up an electron and give up a proton? So you can do this in a few ways. You can first give up an electron. You make the cation radical. That proton could fall off, proton transfer to make the neutral radical. Alternatively, maybe that proton instead wants to come off first, potentially, form the anion. It's still a closed shell species with paired electrons, and then followed by electron transfer to form the neutral radical. Third possible way is both of these happen at the same time. So you have some concerted proton-coupled electron transfer reaction where the proton and electron, there's a coupled sort of motion or movement to eliminate both of them, and therefore you can have them occur simultaneously. Turns out for tryptophan, this is ET followed by PT. So in nature, you see both the cation radical and the neutral radical. You never observe the anion closed shell, and really the reason is thermodynamics. To remove this proton is actually quite expensive in terms of energy. The pKa is very high. It's around 17, 18, 19. And so it's very difficult to actually remove the proton and still hold on to the electron. So these are the two species that are observed, and it has been proposed that it's ET followed by PT. Okay, so our big question here is, we know that tryptophan can be oxidized. We know that it forms a radical. It's in nature. Both the neutral and cation radicals have been observed. And really the big question is, how does the protein control the reactivity of the radical such that it can utilize tryptophan as either a hopping spot or a terminal place for electron transfer? So today, those are the questions. We're focusing on ET and PT, so electron transfer and proton transfer, and we're going to be talking about some isotopes to probe the processes. Will be discussions about singlet, lots of discussion about doublet, because that's what the radical is. I'm not sure how much I'll get on the triplets, but triplet turns out to be an important player, as I've described for at least in Cryptochrome. So what do we need to do? What are we doing in the lab? First, we got to make the radical in a protein. Not such a trivial thing to do because radicals are reactive. It's got to be a very deoxygenated environment. Once we make it, we got to characterize the radical. And in particular, the focus is to characterize the electron transfer and proton transfer efficiencies. Then we get to do more fun work, which is what are the mechanisms, and can we then now create mutants that allow us to enhance whatever it is that we want to do. So that's the roadmap. So in order to make it, we need a model protein. All those enzymes that I showed you before in nature, turns out those are quite difficult to work with in terms of expression, being able to make mutants, the stability. And so if you could have a model protein that allows you to generate a tryptophan radical, that certainly is valuable for the research that we'd like to do, really fundamental studies. So we moved to Azurin. Azurin is a system that's been used by quite a few groups for different types of questions. It's a relatively small protein found in bacteria. It's about it's 128 amino acids, or about 14,000 is its molecular weight. Its goal, its whole purpose for existing, is to move electrons from point A to point B. So it is an electron shuttle protein. It's very likely involved in the nitrate degradation pathway. So there's some ideas about all the other functions that Azurin has, but the dominant one is to reduce nitrate to dinitrogen. So it's kind of the opposite of nitrogen fixation. So it's certainly an important part of the nitrogen cycle. Here is Azurin here. This is the copper center that I've shown, and it's got 5 ligands around it, sort of a distorted geometry. I'm doing just a blow up here of the ligands that are surrounding that copper center. And I show this because Azurin, sort of is how it has earned its name, has a very beautiful blue color. And the blue color arises from a ligand to metal charge transfer from a sulfur here in cysteine to the copper. Very strong absorption at 628 nanometers, which is in the red, giving rise to the blue color of the protein. And this becomes important as we do these experiments. Why are we using Azurin? It's a very stable protein. And by that, I mean it has a very difficult to unfold, so we don't worry about it unfolding. It's pretty easy to express, so we can make different mutants. Crystal structures are known. In fact, we've crystallized it ourselves, so it's fairly easy to crystallize. Quite a bit of prior studies. And it has a single tryptophan residue that has actually quite famous, I'm going to say famous because they're unique spectroscopic properties. Number one, its fluorescence is highly blue shifted. So if I'm showing you here, and I'm sorry, it's very small, I hope you can see this. Here's the absorption spectrum in red. This is in the UV region. The fluorescence spectrum is very featured. If you take model compound of tryptophan from a bottle, its fluorescence is typically around 360, but you see for Azurin, it's very blue shifted at around 307. Why is it so blue shifted and has so many features here? These are vibronic features. It's because that tryptophan is in a really rigid buried pocket. So that becomes important for the stability of the radical. So what are we doing with it? We take Azurin. This is wild type. Wild type means nothing's been modified. This is what it looks like in nature. Single tryptophan, has 2 tyrosine residues. I am highlighting the tyrosines because tyrosines, as I showed in a few slides ago, are also redox active. We want to only study tryptophan. It turns out tyrosine participates in redox along with tryptophan. So the first step we do is we mutate the tyrosine into phenylalanines. So in this particular mutation, which we call azurin 48W, we keep the native tryptophan, convert the two tyrosines to phenylalanines. Phenylalanine is not redox active, and so now we've sort of made silent these two amino acids by converting them to something else. The next step we do is we have to remove this copper center for some of our experiments. Why? Because copper is also redox active. And I showed you the blue color of the ligand to metal charge transfer because the copper 2 to copper 1 is pretty straightforward chemistry. And in fact, that is what this protein does. So we can convert copper, take that out, and replace it with a zinc. Why zinc? Zinc is a D10 metal. It has no transitions. So you lose the color and it can no longer participate in any redox chemistry itself. We also have a different variant where we just remove the metal center altogether, and this is called apoazurin, which has no metal. All of these still we're keeping the single tryptophan residue. If I look at the absorption spectrum now, it's as expected. I already showed you the UV region of the absorption, but here's that very nice ligand to metal charge transfer of cysteine to copper here at 628. If we make the zinc or the apo variant, that ligand to metal charge transfer is shut off, and therefore we see no absorption here in the visible region. So the protein goes from a deep blue color to basically clear. But the reason for making these mutations is really to isolate tryptophan and make sure we understand that the radical is from the tryptophan moiety. So how do we generate the radical in the protein? So this is photo-induced electron transfer. In order to have electron transfer, you got to have a donor and an acceptor. Something has to give up an electron, something has to accept it. So what is accepting an electron? We can add small molecules such as cobalt or ruthenium, which we have done. How does this work? Here's our zinc protein with the single tryptophan at position 48, no tyrosine. Photoexcite with 280 nanometer light. An electron is then ejected and accepted. Cobalt 3 becomes cobalt 2. We don't have to use cobalt. We can use anything we want. In our case, we actually use the copper variant of azurin. Why do we do this? One, it forms a dimer, so it actually does some nice chemistry. But really, the reason we're using a protein as the acceptor is because the copper 2 band has this nice color, which gives us a marker of the electron transfer. And you'll see what we mean when I show you the graphs in a second. Do we know that the electron is actually getting from W48 to the copper center? We have a hypothesis as to what the electron transfer path is. It's a viable path, and so we do believe and we do see this reduction. So it's a nice system that works. Okay, so how does this work when we're trying to make the radical? Here's the protein, zinc substituted, some acceptor in solution. It could be cobalt, it could be copper. Photoexcite with 292 nanometers, which is UV. Create the excited state. There's still an acceptor nearby. Electron transfer will result in the cation radical. Proton transfer will follow and will result in the neutral radical. We have a reduced acceptor here, and then we have the proton come off. If we can isolate these two species, we can study the neutral radical or the cation radical. The neutral radical is actually quite straightforward to study because it lives for quite a long time in the protein. So this is hours. So it takes over 3 hours for it to decay. So it actually allows us to do, and technically it's time-resolved experiments, but it's on the order of hours. So we can do quite a bit of spectroscopy on the neutral radical. Cation radical is more difficult. So it has actually been something we've been trying to study directly. And so we've still working on that, but we do have some data on some model compounds. What do we do with this? Quite a bit. We want a library of spectroscopic signatures so we can understand the radical. This had not existed, prior to us getting involved. So we analyzed the absorption spectrum, the EPR spectrum, of course, the Resonance Raman spectrum. And I'm going to spend quite a bit of time on the Resonance Raman component. So I'll talk a little bit more about that here, which is a tool that our group uses quite extensively. So resonance Raman spectroscopy is simply a form of vibrational spectroscopy, but it's based on scattering, inelastic scattering, not absorption. It's quite selective and sensitive, and it really reports on bonds, so molecular structure. Is it a single bond? Is it a double bond? Is it a CH bond, a CC bond? How does that bond, what is the vibration in different environments? So whether it's polar, non-polar, also affects the structure. And of course, you can also get information on the dynamics. So just to make sure we're all on the same page in terms of what resonance Raman scattering is, as an inelastic scattering process. So what I've drawn here are the potential energy curves. These are just sort of cartoons that I've drawn, harmonic surfaces, ground state. So this will be the S0 state of tryptophan. I showed you the absorption spectra of tryptophan. It actually has a degenerate pair of LA-LB. This is the 280 absorption. It has an absorption at BB, which is the about 220 nanometers. In resonance Raman, there's an incident photon, in this case, resonant with an electronic transition, and then scattered light that is lower in energy than the incident by an amount of a vibrational quantum. And therein is how we get information about the vibrational structure. And this is, you can compare this to infrared absorption, which many people are familiar with, which is simply a V equals 0 to 1 transition direct absorption. And many people think of resonance Raman or Raman as a complement. The selection rules are different. Resonance Raman itself is quite powerful because if I were to look at the expression, so this is now the sum over states expression. Here's the cross section for resonance Raman scattering for a given mode. This could be for any mode that we're looking at. And this cross section simply tells you how likely it is to get this type of scattering event. So it depends on the energies of the excitation as well as the scattered light, depends on the overlap of the wave functions here, which is the numerator. So V represents the intermediate state. And then the denominator has the energy term where once the energy of the laser is resonant with the excitation, with an electronic transition, the denominator goes to 0, resonance Raman cross-section goes up, and therefore, by picking the correct wavelength, you can tune the signal from your absorbing species by up to 6 orders of magnitude. Why am I going through all this detail to tell you this? It's because when you have a protein sample, which is pretty low concentration, so maybe 10 to the minus 6 molar, in water, which is 55 molar, we only want spectra of our molecule of interest. And by using resonance Raman, by tuning this wavelength, we're able to do that. Our group, many other groups, certainly this is not, this is work from Raman spectroscopy, of course, got the Nobel Prize. I think it was the late 20s or maybe early 30s. So it's a very rich technique, lots of advancements. I can just show you an example. If I take a peptide that has tryptophan, so the indole side chain and the backbone, if you excite where the tryptophan absorbs versus where the backbone absorbs, you'll get 2 different spectra. So with 230 nanometer excitation, which is absorbed by the tryptophan, you get a spectrum that might look like this. And if you move it 20 nanometers away, you get resonant with the backbone and you'll get a spectrum that looks different. This spectrum up here on the backbone tells you whether it's alpha helix or beta sheet. So it tells you about the secondary structure. This one is telling you specifically what is the environment of a side chain, the side chain being tryptophan. So this was for peptide melatonin, which is the venom in honeybees. When you get stung by a bee, this is the peptide that is causing the lysis. And so just an illustration of resonance Raman and the power of resonance Raman to give us the type of information that we're interested in. What does a resonance Raman spectrum of tryptophan look like? Again, many others have studied this. Here's that Raman shift, which is the difference in energy between scattered and incident light. Here's the intensity of basically number of photons for that particular mode. All tryptophan normal modes are labeled W blank. Blank is just a number from 1 through 18. It's the in-plane normal modes here. And I'm just showing you some of the, these are just from Gaussian calculations that show you the displacements of the vibrations. We and others have spent quite a bit of time understanding the spectrum. For example, this mode here is very sensitive to the structure. There's what's called a Fermi Doublet, which is very sensitive to the hydrophobicity as well as pi-pi interactions. Hydrogen bonding markers, W17, W10, and some other markers here for cation pi, as well as just local environment. So a lot of information that we obtain from Resonance Raman. So let's go to what we get for our molecules. So here is, because tryptophan absorbs in the UV, this is UV Resonance Raman. I just showed you this spectrum. This is what I just showed on the previous slide. If you oxidize it by removing an electron, you're left with a cation radical here. Here is now the Resonance Raman spectrum of that. This is visible because the cation radical absorbs, this doublet absorbs in the visible region. Remove a proton, you're left with a neutral radical, and here is its Resonance Raman spectrum. So this notation of W1 through W18, we mapped on to the cation radical, we mapped on to the neutral radical, just to show how these normal modes are changing in frequency, actually as well as displacements, and what makes them up as a function of sort of the species. These two here, the cation radical, neutral radical, the way they're drawn is based on the spin densities. So coming from EPR work from other group, as well as from our group here, where we're showing, spin density is largely here for the neutral, as well as, and then distributed here for the cation. And the type of information we got, again, the whole goal of this is so that we have spectroscopic markers for when we're trying to study, do we see a cation radical or a neutral radical? And we can look at something like the W3 marker, which is sensitive to the structure. That kind of reflects the C2, C3 stretch. So from a local mode picture, the C2, C3 stretch, which I've circled in green. In the cation radical, it stays, basically retains its double bond character. When you get to the neutral radical, it downshifts about 200 wave numbers and becomes more single bond-like, showing that this is really the dominant resonance structure of the molecule. So it's really a combination of maybe what you've learned and with sort of basics of vibrational frequencies and structures, but our ability to do this and do the assignments helps us, then sets us up for these future studies, in particular for time-resolved work. Well, let's go back to our theme here, which is electron and proton transfer. I've already shown you this, where we can have electron transfer, proton transfer. If we can quantify the amount of reduction of the acceptor, we can extract a yield for electron transfer, which we're going to denote as phi-ET. This we've defined as the probability for an ET event per photon absorbed. If we go to the other side here, we can now look at the neutral radical and analogously define the yield for forming the neutral radical, which we call phi-rad, probability to generate the neutral radical per photon absorbed. The only way we're going to make the radical is if there's ET and PT. So now we can extract a proton transfer efficiency. We're going to call this phi-PT. This is now the probability for a proton transfer event per electron transfer event. So it's a little bit of a different definition, so I'm going to call this the efficiency. But the product of these two gives you the radical. And so what we're after is understanding these efficiencies and yields. Now the reason why we use a copper azurine for the acceptor becomes important, because I need a marker for the reduction of the acceptor. If copper 2 is the acceptor, it becomes copper 1. Copper 1 is a D10 metal, it becomes clear. This ligand to metal charge transfer disappears. We know the extinction coefficient well, so there's a bleach of the acceptor, there's a rise of the neutral radical. I plot the growth of these as a function of photolysis time here, extract the radical quantum yield, and extract the electron transfer quantum yields. Now I take that number, so here is the zinc protein with a single tryptophan. the radical yield is about 0.23, which means per photon absorbed, it's basically a 23% chance the radical is going to be made. I can then look at the electron transfer yield, and these should be higher or equal to the radical yield. In this case, it's 0.25. I know that the product of ET and PT must give radical, so I can extract the proton transfer efficiency. What these numbers tell us is, first, that the efficiency for proton transfer is very close to 1. In other words, every time an electron is transferred, a proton is basically also lost. The error here essentially says it's nearly unity, and so it's a very high yield. We were a little bit surprised to see the proton efficiency be as high as it is, because we know that the cation radical has a finite lifetime. In other words, during the lifetime of the cation radical, it's possible it could have been quenched by a different reductant or something else, but it doesn't seem to be happening. So one might wonder then, is it truly a sequential set of steps, or is it a concerted? And so we'll get to that question. But this yield is quite high because just nada itself, for L-trip, we can eject an electron, but that yield tends to be lower, around 10% or 15%. So the protein somehow is able to allow the electron transfer yield to be higher. We wanted to understand this better, so we moved to isotopes. And isotopes are really valuable experiments for proton transfer chemistry, and it turns out also for electron transfer chemistry. So what are the isotopes? I'm going to call this native. Hydrogens on all the rings. This exchangeable proton is also hydrogen. I already showed you the quantum yield to make the radical is 0.23, and here's the absorption spectrum that led to that quantum yield. We can investigate what is called the primary isotope effect. That's basically exchanging out this NH for an ND. So now we have a deuterium, a heavier atom, but the ring hydrogens remain hydrogen. That has a reduced formation yield for the neutral radical by a factor of about 1.4 compared to the native. We can then investigate the secondary isotope effect, and what that means is we keep this proton NH, but we exchange these. We actually don't exchange. It's an unnatural amino acids because these are not exchangeable protons from hydrogens to deuterium, and so now we have something that allows us to probe the secondary isotope effect. The radical is still formed in this D5 variant, so I'll refer to this one as D5, this one as ND, and its radical quantum yield is also reduced about a factor of 1.4x. Here are those numbers that I just told you, and you can ask yourself, or we asked, why do these radical yields vary? Because all we've really done here is change the isotope. The isotope in principle should not affect the electronic structure, because to form a radical, the electron is ejected. Of course, the proton must come off. So we have to look at the individual parts. Let's look at the electron transfer yield. It turns out between the native and the ND, the primary isotope, the electron transfer yield is actually not very different. The D5 is where the number now gets reduced. And so why do these electron transfer yields vary? Why do we have basically unchanged for ND, but then it changes for D5? We don't think the protein structure is changing among these three. We don't think there's an excited state lifetime argument. So we've looked at fluorescence and we've investigated that. We do not think that that's the reason why. We think instead it reflects that vibrations play a role in electron transfer efficiency. Again, this is something that's known, and this is allowing us to really investigate it further. So what is the role of vibrations in electron transfer? Because you might think, well, if you're ejecting an electron, what do the nuclei have to do with it? And of course, the nuclei are very important. And if we just look at sort of a Marcus theory expression, there are both electronic and nuclear terms here, electronic term, and here's a nuclear term that I'm highlighting here in blue. That's the reorganization energy. Very simply, reorganization energy refers to the energy that's associated with nuclear relaxation to the new equilibrium geometry of the now charge separated products. So the donor, which normally was neutral, has now become charged, and so the nuclear geometry has to adjust. Both the solvent, which is the outer sphere, as well as the molecule itself, which is the inner sphere. This is a part that depends on the isotopes. So now if you have isotopes, that lambda I is going to change. And therefore, that explains why the D5 has a different electron transfer yield. Resonance Raman turns out to be quite powerful for these types of numbers. And if I transition just a tiny bit, a little segue, if you have a photo-induced charge-separated state, so this is not necessarily the case we have here, but if you do, Resonance Raman can directly report these inner sphere reorganization energies. Why? Because the Resonance Raman intensities depend on the frequency and this thing called a delta. The delta is what tells you this change in geometry upon the excitation, somewhat analogous to this change in geometry upon charge transfer. So what these data are telling us is that if we look at the Resonance Raman spectra of ND versus native, if you recall, those had the same electron transfer yields. Look at the Raman spectra, basically the same. There's a shift of the W17 mode as expected, because that's the NH, it's indicative of hydrogen bonding and the NH displacement. Now we bring up the UV resonance Raman spectrum of the per deuterated D5. Its Raman spectrum actually looks quite different from native, and it also has a different electron transfer yield. And in fact, if we look at, for example, the two biggest peaks in the Raman spectrum, 16 and 18, we see that the intensities have kind of flipped on the D5 variant. All of a sudden, 16 is bigger and 18 is smaller. These modes have different relative intensities. If we go back to what I just said about reorganization energy for a photo-induced process, if I look, for example, at the W18 mode, now first I want to state that a mode in an H5 molecule compared to a D5, those do not necessarily match up. You will not get 100% identical mode similarity. So this one turns out to have less than 40% mode similarity. So this is for us doing the vibrational mode analysis. Therefore, the total internal reorganization energy is expected to be different. If I draw it in terms of the displacement, so here's the displacement of that W18 mode for the native and the per deuterated. In the native, it has a larger intensity, which means its geometry is more different in the excited versus ground states. So it's a little bit, it's a deep topic, and I know I'm not going into it enough detail. I'm bringing this up to highlight that the Resonance Raman data are consistent with expecting D5 and H5 to have different electron transfer yield. So we can ascribe that to the normal modes. We can then tabulate these isotope effects, which we'll call primary and secondary for electron transfer. There basically is no primary isotope effect, meaning ND and NH have essentially the same result. So there's no heavy isotope effect or no effect of the 0-point energy, which is typically what you think of for D versus H. However, there is a secondary isotope effect, which we believe is the effect of vibrations. Let's go to the proton transfer part here. What is its efficiency? Well, native compared to D5, which also has an NH, basically no change. But this middle number, all of a sudden, we put an ND on there, its proton transfer efficiency goes down, only for ND. I think this explanation is a little bit more intuitive. Well, I put a heavier atom there, and so the heavier atom is going to be more difficult to remove. And sure enough, this is sort of falls along the lines of a classical isotope effect. You've got a lower 0-point energy for ND versus NH, and so this gives rise to this primary isotope effect, no secondary isotope effect for proton transfer, which means there's really no effect of the vibrations. Or if there is an effect, it's modest, it's very small compared to the actual NH versus ND. So we believe we understand those efficiencies pretty well. We are interested, of course, in the kinetics. So not only do we want to know the yields, but how fast are these things taking place? This is another topic we could spend a lot of time on, and I'm just going to give you 2 slides of the work we've done. If we now take the metal off, so it's apoazurin, we do this because it turns out apoazurin is not as stable in terms of making the radical. No metal. So we add an electron acceptor. In this case, we acted cobalt. We did the same chemistry, but now we get a slightly different result. Here's the absorption at 514 nanometers, which is where the radical absorbs. No UV light on. We turn it on. We make radical, goes up. We turn it off. The radical is gone. This is quite a contrast when we had zinc in there because it persisted for 7 hours. When all the metals, no metal in the center, it only persists for about 1 minute, less than a minute. And if we look at the absorption spectra, that confirms the assignment because at A, before any light is on, we see essentially this kind of this is sort of a background here, A. We turn the UV on and measure an absorption spectrum. It looks like the radical. We turn it off, and again, the radical disappears. And we can compare these little bumps here to what I showed you for the tryptophan radical that we know. It's the same set of bumps, it just doesn't persist. So what do? We take these rises and decays as a function of excitation power. So we've actually done about 8 of these. I'm just showing you a handful of them. We now set up a kinetic model. We have an excitation rate. What are we trying to get? What's the goal of our kinetic model? I want to know the rate constant for ET and the rate constant for deprotonation. There's some other terms in there, but really I just want to show you the bottom line is that the rate constant for ET and deprotonation, microseconds to sub-microseconds, there's no surprises in these numbers. This is the typical type of numbers one would see for this type of chemistry. So our modeling seems to be working, and while this is not we're not directly monitoring the cation radical, we think that we're understanding these rates. So the last couple minutes here, I'm just want to tell you about the mechanisms for ET and PT, because this is like the big picture. We want to know how it is that the electron's getting out, how it is that the proton's getting out. I've already shown you that the vibrations affect the electron transfer efficiency, and we have a hypothesis that how is the proton getting out of this very rigid pocket? This tryptophan 48 points, has an NH, points to phenylalanine, which is right here. Phenylalanine is near a threonine, and we believe threonine affects PT efficiency. How's this going to work? And why is this important? Let's go back to the motivation. I started out telling you about TRIP48, about electron transfer, and how nature uses intermediates to enhance long-range electron transfer. If nature wants to use tryptophan, what has to exist? You got to have ET that's going to work, and you have to stabilize the cation radical. In other words, don't lose a proton. Why is this? Let's say we have a donor, which is a pink box, and an electron has to be transferred a long range over to this green triangle. So nature wants to send an electron over a long distance. Nature's decided to put a tryptophan in there as an intermediate. So great. Electron transfer, success. We've got charge separate. The charge is transferred from the green triangle to the pink box. What if you do electron transfer and tryptophan loses a proton? Now we've got a kinetic barrier because in order to fill the hole, it's also got to get a proton. So I call this a dead end because now it's going to be a difficult, much more difficult chemistry to achieve. So you really want to stabilize this cation radical. So in order to do this, what we've done is we've taken threonine, which looks like this. It has an OH group, so we believe this is very important to the solvent-exposed nature. We made a mutant, which we call closed. We converted this OH, put in a valine, which has a methyl group. What happened? As expected, when we closed this pocket, the proton transfer yield was reduced, which means it stabilized the cation radical. We did the reverse. We made an open mutant, which is alanine, and there was no change to the proton transfer yield. It's a little bit consistent with what we expect because the proton transfer yield is already high at 92.92. So it's hard to get higher than that, but we were glad to see this result. I have a summary here, which I will skip very quickly in the future that topics we're working on. And just for the sake of time, again, thank you all for being here. Really value being able to share our science like this in an international manner. And of course, very importantly, the students, again, who have done all the work and the funding agencies that made this possible. So thank you very much.
Thank you, Judy, can you hear me?
Yes.
Yes. Thank you so much for the very nice talk. I have to admit that many of us probably come from a slightly more quant background, so it might be it's been really interesting to see that some basic concepts that we use on our everyday basis can also be applied so heavily on more applied and biochemical environment. Maybe I can start asking some very basic questions. So in all the basic in your presentation, you never mentioned essentially the role of temperature. So I was wondering, I guess that all your basically the results that you showed are relate to room temperature, but correct me if I'm wrong. And I wanted to understand also what is the role of temperature in both in the electron and proton transfer processes.
So excellent question. And our experiments are all done at room temperature, perhaps a little bit elevated, and that's because of the protein. So the limitations of the protein restrict us to the place where we know the dynamics are optimized. So we have tried low temperature, but the protein doesn't seem to be very stable. So even getting to 10 or 15, even too elevated is also the protein's not as stable. For ET, temperature is a wonderful parameter and very important. Since we're trying to understand it in the protein, we don't have as much leeway in terms of temperature. So we don't have a conclusion about the temperature effects on the protein again. We have kept it at room temperature or perhaps slightly elevated. But your question is a really relevant one in terms of just the fundamentals of ET. There is a strong temperature dependence for ET in general. Right.
We have a question from Tiago. He's asking, could you please explain why Z and azurin has such longer lifetime than apoazurin?
Yeah, no, this is a wonderful question. Why do we think that? So that is from this slide here. And the reason is because the dynamics. So let me put this here. Here is the bottom line. Apoazurin, because it does not have a metal center, those ligands actually can move around a little bit more and you can get water in that cavity. So there are structures, and in fact, there's NMR structure that shows that the ligands can sort of move in and out. You have more than one structure that is possible, and this hole from lack of metal makes the entire sort of pocket less rigid. And we think the rigidity is very important for the lifetime of the radical. And so for APO, and it works for us because we wanted these kinetics. We could not see this in zinc azurin. We could not model that. That's an input in our kinetic model.
Okay.
It was not planned that way in the sense that it was a surprise to us too, as many experiments often are.
Yes. Maybe I have another question, Judy. It's probably somewhat related to what you asked. I see that also Sandra wants to ask. Maybe I can just finish this question, Sandra, and then I will let you ask your question. I was curious about these processes, so I got interested recently more from the quant math and atomic physics background about the role of the interplay of disorder and temperature effects. So basically, the fact that all these processes, which are essentially very basic physical processes, take place in the presence of an external environment. Mm-hmm. So is it easy to characterize essentially the environment and what is the role of the environment in the experiments that you basically described your presentation?
So another excellent question. Fortunately or unfortunately, I think it's fortunate, the protein has a really well-defined environment. So for us, the fact that the protein structure is known so well, the dynamics are, whether you're doing simulations or looking at NMR studies, those we can at least interrogate. So I think that makes this problem, in some sense, more straightforward. The environment, the dynamics of the environment, and the actual chemical environment. So is there a hydrogen bonding partner? Is it hydrophobic? Both of those, I think, are equally important. And this relates to why APO is so unstable. I think the dynamics of that pocket are just more enhanced in the absence of this metal, and therefore destabilizing this radical. So that helps with us. There's, of course, the area outside the protein, all of the water and such. There's no water in the pocket where W48 is, but the water here on the outside will obviously affect the environment of the protein. But really, I think it is the key thing about why this works for this protein, I believe, is because that local environment right around W48 is optimized for this. So I think that is why the system works well. We are doing many mutations, well, trying to do, where we can, for example, mutate out this environment to change it to, let's say, less hydrophobic. What is the effect? Will it stabilize a charged intermediate, for example? Because this is a big question. If we want to stabilize this right here, we want a protein pocket that can stabilize a charge. So then, fortunately, with mutagenesis, we can approach that with making mutations.
Right. Thank you.
Hello.
Thank you. Please, we have another question. Please go ahead.
Okay. So first, let me thank you for the talk. Like it's pretty much, although I don't think I understood everything, but I have a very basic question. At the very beginning, you showed a graph where a very large range of tunneling time for as a function of the distance. My question is tunneling the only mechanism that is responsible for this kind of transfer, electron transfer, or are there other mechanisms to?
So you're right. The timetable that I showed you is the tunneling timetable. In our, when I showed you these results of the proton transfer efficiency, let me just get here. We believe that this is really not a quantum effect in terms of the tunneling, that we believe you can just explain it in terms of a classical barrier and needing to go over the barrier. So the language of, do we have to say it's always tunneling versus can you describe it as a classical viewpoint? I'm using a little bit of a classical picture here, but clearly if we're thinking about electrons moving through, the quantum effects are non-trivial. They have to be real. So I don't know if I'm answering your question properly. Generally, tunneling is the way that this is being described, but I think it is valuable to still keep in mind that classical pictures can be used to explain, for example, isotope effects.
Okay, thank you.
Okay.
Please, Sandra.
Okay. First, thank you for the very nice talk. And I have a curiosity. Do you think that can be used in nonlinear Raman technique to this kind of studies?
So a non-linear Raman technique. So in terms of, are you talking sort of like stimulated Raman or cars or any of the other ones?
Yes.
Sure. No, there's no reason you could not use something like cars or SIRS or any of the other tools. We don't need to because Resonance Raman is working well. This is, in terms of Raman, this is actually a pretty easy problem because it's got a beautiful color. There's not very much fluorescence. So if we needed to do surface enhanced because, resonance just by tuning the laser was not successful, absolutely, you certainly could. The challenge with a protein, though, is we like to keep it in solution and not fixed on a surface, not adsorbed onto, like, say, a silver nanoparticle. So if we can avoid these things, I think it's better in terms of isolating the electron transfer chemistry we're interested in. So, but there's no reason you could not. You certainly could use a different type of Raman.
Okay, thank you.
Judy, maybe another final question, which basically stems from out of curiosity regarding the type of application that you described at the very beginning, which is to butterflies.
Mm-hmm.
And you said that these butterflies essentially feel the external magnetic field or the Earth magnetic field. I was wondering, in these experimental approaches that you mentioned, you are talking usually about spectroscopic measurements. I was wondering, how do these results affect, or how are these results affected by an external magnetic field?
Oh, so
is it a relevant question, or
yeah, no, it's an excellent
does it make
question. In
sense?
fact, one of my students has tried to measure the yields in the presence of a magnetic field to see if the yields are different. You know, in terms of the butterflies, it's the interaction between two radicals that is what is proposed to be sensitive to the field. We have tried to do you know, magnetic field effects on the radical quantum yield. They were not successful at the time that we tried them. But I think your question is perhaps a little more related to the mechanism by which the butterflies are able to sense, you know, magnetoreception. But I think the effect of magnetic field will be a really interesting, you know, path in terms of this entire chemistry, since the interaction, especially if we can get pairs of radicals, we can study cryptochrome itself. So it's a fun question.
Okay. Thank you so much, then, Judy, for the presentation, and I hope that our guests also enjoyed it. And I hope that at some point we could also meet in person. And thank you so much.
No, thank you, and thank everybody for watching. I really had fun presenting it. I wish we can meet in person, and I wish you all well, and I hope our paths will cross again. Thank you again.
Thank you so much, Judy.
Bye-bye.
Have a good day. Bye-bye.
Bye.
You. Bye-bye.
Bye.